Welcome to part 2 of our urinary system lecture. This time we're going to talk about the nephrons, which are in the kidneys, and they're what's doing all the work of filtering the blood and creating the urine. This lecture is going to match up with page 521 to 524 of your textbook that talks about the nephrons and then talks about the steps of urine formation. All right, just to refresh from where we left off, we talked about the structure of a whole kidney last time. And we talked about that cortex layer and then these sort of triangular looking things that were called the renal medulla or the renal pyramids. And they drain into this renal pelvis where all the urine is collected and then goes out to our bladder. But in this renal medulla and these triangle looking things are millions and millions of little structures called nephrons, and that's what we see over here. And those are where all the filtration is going to happen and the urine's actually going to get formed. Okay, so what is a nephron? That's what's pictured over here. And they are considered the structural and functional unit of the kidney. And what that means is if we got down to the smallest working bits of the kidney, this is what it is. Once you take a nephron apart, it's not going to work anymore. There's over 1 million in each of our kidneys, so we have over 2 million in our body, and they're all working constantly. And there's two main parts. There's the vascular part, which we know from our cardiovascular system is the veins and the arteries, the things that carry the blood. And then there's the renal part, and we know from last lecture, renal means kidney. So this is the part that's carrying the urine. So basically, a nephron is the point at which blood becomes urine. So the two vascular parts you're really going to need to know are this blob here called the glomerulus. And glom means basically a knot or a circle of something. So this glomerulus is a knot of just little capillaries. So here comes the blood originally from our heart down our aorta, and then it branches off into the renal artery, and then it's going to branch off smaller and smaller as it goes through the kidney, and then tiny little arteries are going to get to each individual nephron. And so here comes the blood in, it hasn't been filtered at all, and it's going to knot up right here. And this is where the filtration is going to happen. And then once that blood has been filtered, it goes on, and we'll talk about what it becomes in a little bit, but these are called the peritubular, meaning they're wrapped around the outside of these tubes, capillaries, and they're going to reabsorb the blood once it's been filtered. So that's the vascular part. The second part is our renal part, and that's this yellow tubing you see here. And that's once the fluids have been filtered out of the blood, now they're going to travel along these tubules in the process of becoming urine. So here's the renal tubule, and it's traveling along, and things are being added to it and absorbed out of it until the urine contents are just right. And then when it's all done, it's going to dump into this collecting duct, and this collecting duct is going to drain into our renal pelvis and then eventually to the bladder to be excreted out. So we're going to talk about each of these parts in a little more detail. So here's our glomerulus again. Like we said, it's just a knot of little capillaries. And it's being hugged by this Bowman's capsule. Now, in your book and in your diagram packet, sometimes it's called the glomerular capsule because it wraps around the glomerulus. But it's also named for Bowman, a scientist who came up with it. So either one you can use. They're interchangeable. In this lecture, I'm going to call it Bowman's capsule. Our glomerulus is fed and drained by arterioles, things that are under pressure, things that are carrying oxygenated blood. This is rare. Most things are fed by an artery and then drained by a vein. But here it's just sort of making a stop along the way. And so because these are all arteries, it's under very high pressure. And that's important. We want the pressure to be forcing this liquid from the blood plasma, out so that it can be filtered and become urine. Remember from our nervous system, afferent means going in. Afferent with an A was our nerves that were carried into our brain. And efferent means exiting or leaving. So we have an afferent arteriole coming in, blood's being forced out to be filtered, and then as it's leaving, we call it efferent. Okay, so once it's forced out, it enters the renal tubule. And here, I like this picture because it shows the glomerulus, but then it sort of peeled away all of those veins and arteries that were wrapped around, the capillary beds that were wrapped around. And so you can just see that we sort of have this twisting and turning tubule, and then it makes a loop here, then it twists and turns again, and then it dumps into the collecting duct. So this starts with Bowman's capsule. That's this thing here that sort of looks like a fist that's hugging the glomerulus. And this is the site of the actual filtration that starts off the whole process. Once blood is forced out by that high pressure, so the plasma and things that are dissolved in it are forced out, it's collected up and caught by Bowman's capsule. It's going to move through what's called convoluted tubules. And they look convoluted. They're all twisted and coiled. Okay. There's a distal and a proximal, and I probably should have listed those in the other order. So the proximal convoluted tubule is the first one it goes through. Proximal means closest. Then it drops through this loop of Henle, which we'll get to in a second. And then there's a distal convoluted tubule, and then it dumps into the collecting duct. And again, this is the loop of Henle where it sort of makes a U-turn and leaves. Now, think about what you know about structure and function. Why do you think that this is so twisted and coiled and then loops about and then twisted and coiled again? Why can't it just be nice and even and stretched out and not coiled up like this? I'll give you a second to think about that. Okay, one of the reasons is space. We have about 1 million of these nephrons in one tiny little kidney, and we saw the other day our kidney's not that big. It's about the size of a bar of soap. We got to fit 1 million of these in there. We can't afford for them to all be stretched out. So if they were going to be stretched out nice and straight, they would have to be much shorter. What do you think the problem is with this renal tubule being shorter? Think about what do you think is happening there that it needs so much length. And I'll give you a hint that we've seen this in our digestive system too, maybe in the small intestine. Okay, the reason we want so much length and therefore need to coil it up and twist it up to make it fit is because what's happening here is things are being reabsorbed out of the urine into the blood. We don't want anything that we can still use to be peed out if we can reabsorb it into the blood and use it. Things like blood sugar. And so by making this nice and twisted, we can fit a lot of length in a very little space and give it plenty of time to reabsorb what it needs, kind of similar to the small intestine absorbing nutrients. Okay, so we already talked about the glomerulus, which was that knot of capillaries that drops off the blood under high pressure. But where does that efferent arteriole go? It becomes the peritubular capillaries. And we keep seeing this word peri. It means outer covering or wrapped around the outside. And then the tubular refers to it's wrapped around the outside of the renal tubule. Capillaries, we know, are just tiny little blood vessels. Okay, so this is the capillary bed that is all wrapped around the renal tubule here. And it arises or starts from the efferent arteriole. So here comes our renal artery, here comes the afferent arteriole under nice high pressure into the glomerulus, forces some of the blood plasma out to be filtered, and then it takes what's left over and it leaves through the efferent, and now this is going to branch off into the spidery, webby looking capillary bed. These are under low pressure. So once we've gotten past the glomerulus, that pressure is going to drop. They're under very low pressure, and they're very porous, meaning they've got lots of little holes in them. This is very good because their job is to absorb things, and if they were under high pressure, they'd be forcing stuff out, not letting stuff in. Oh, sorry. So those pores and the fact that there's not a lot of pressure going on means things can easily be absorbed back into them. And then ultimately, once all that reabsorption's done, they're going to come back together. The web's going to sort of condense up, and it's going to leave through those renal veins and go back to the heart and start again. Okay, so how does urine get formed through all this? Now we know about the structures. Let's talk about the 3 major steps, and these are described very well in your book. The steps are glomerular filtration, tubular reabsorption, and tubular secretion. We're going to talk about each of these one at a time. Glomerular filtration is the step of getting these things out of the blood into the nephron so that they can be filtered and adjusted, and we can do that constant balancing act of homeostasis and get our blood contents just how we want them and pee out the rest. So we already talked about this. That high pressure of our afferent arteriole is going to force things out of the capillary knot there, out of the glomerulus, into our Bowman's capsule so that it can continue through the tubules. And the things that get forced out are water from our blood plasma and the solutes that are in it. Anything smaller, though, than blood proteins. So all of our proteins, all of our clotting factors are staying in the blood. The red blood cells, the white blood cells, things that are big, well, relatively big, are going to stay in the blood. We don't want to be forcing our blood cells out. That would be bad. So it's little things like blood sugar, glucose, little amino acids, little ions and salts. This is a non-selective and passive process. Here's what that means. Non-selective means it's not picking and choosing. It's not saying, oh, you're a glucose, okay, you can leave. Oh, you're a protein, okay, you have to stay. Anything that's small enough to fit goes. This is part of the reason that we're then going to need to reabsorb some things, because some things that we would really have preferred to keep are going to get out. So non-selective means if you're small enough to fit through, you're going to go. We're not being picky and choosy. There's nobody standing at the door checking IDs. The passive part means it doesn't require energy. Okay, this is a process that there's already the pressure there. There's the space to fit through. It's just going to go. We don't need any energy to force it, which is good. Think about if we needed ATP every time we had to pee, it'd be a problem. Okay, this is pretty cool. The filtering process, and the reason that things that are too big that we don't want escaping, like proteins in our blood cells, can't fit out is because of these little guys called podocytes. Look at that word. Look at the podo part. Think about where you've seen this prefix before, and hopefully you all know what cytes means. What do you think podocyte means? Okay, hopefully you were thinking that podo means foot. Think about going to a podiatrist. This means foot-like, and cytes, I hope you all know, means cells. So these are foot-like cells. So what happens is our little capillary knots are here, and the outermost layer of the Bowman's capsule, the one that's actually bumping up against these capillaries, are made of these podocytes. And they have normal cell bodies, but then they have these little foot-like extensions that wrap around the capillaries. Think about where we've seen this before. Where have we seen something like this before? Hopefully you're thinking about something in the nervous system. Okay, we've seen these before with our astrocytes that wrapped around the capillaries that feed our central nervous system. That's what forms the blood-brain barrier. Well, these podocytes are very similar. And so what happens is all of these little legs, you can see it in the actual picture of it here, all these little legs sort of intertwine with each other, and they sort of form a sieve. They form this layer that only tiny little things can fit through. And so naturally, only ions and small things fit through, and proteins and blood cells are trapped and stay in the blood. Lastly, having the correct pressure is very important. When pressure gets too high for some reason, there's a blockage or somebody has constantly very high blood pressure, that's going to be a problem because things are going to be forced out, things that shouldn't, and we'll start to see differences in the urine content. Secondly, if the pressure is too low, if your blood pressure is dropped way too low, there's not enough pressure to force things out, and things won't get filtered properly out of the blood. So keeping that correct pressure so that the right things get forced out into the urine is very important. All right, let's move on to step 2, which is tubular reabsorption. So like we said, if the first step is not selective, then some things that we didn't really mean to get rid of are going to move out of the blood. So step 2 is where we're going to be choosy, we're going to be selective, and we're going to reabsorb things we want. So anything our body can still use or still needs to stay in balance is going to get reabsorbed. First and foremost, water. So water is going to be reabsorbed by osmosis. If there are more things dissolved, more substrates dissolved in our blood than in our urine, water is going to come back into the blood and help water those down. Then things like glucose, amino acids, any ions we might need like sodium are going to be reabsorbed, but this is going to be by active transport. This is going to take a little energy. We're going to have to do this intentionally. So what happens is the cells that line those capillaries have what's called transporters, and they're basically little spots that look for whatever it is they are designed to reabsorb. So I tend to think of this as people at the airport waiting for somebody. Maybe it's a limo driver waiting for the person they're supposed to pick up, and you've seen probably videos of people standing there with a sign that say, Mr. Smith. Okay, this is like a little cell that's standing there with a sign that says glucose. If you're glucose, I'm here to pick you up and put you back in the blood. The more transporters there are, the more reabsorption is going to happen. So this is how our body regulates things. We don't want to pee out blood glucose. We can use that to make energy. We can do cell respiration with that. We don't want to get rid of it. So we're going to have a lot of transporters for glucose and hopefully catch all that glucose and not pee it out. We do want to pee out urea. Urea is a waste product. We don't like it. It makes our blood too acidic. We want to get rid of it. So there's very few reasons why we would want to reabsorb it. Therefore, we have very few transporters. Nobody's waiting at the airport for this guy. All right, last step. So now we've forced out things passively and non-selectively. We've just gotten anything that's small enough out through glomerular filtration. Second, we reabsorbed the stuff that we wanted to keep. Most of that was active. We were very choosy. Okay, that was tubular reabsorption. So now they're going back into those little peritubular capillaries we saw. They're going to go back to the heart eventually. Next, we want to sort of have a last minute check, last minute readjustments, sort of a final edit before we can't get anything back. Okay, so certain substances didn't get forced out in the first place. Remember, that was non-selective. We couldn't pick and choose. And so certain things, we still want to get rid of them, but they have to be actively secreted out of the blood and into the urine that's going to get excreted. That's things like hydrogen ions. So this is where we're going to be helping adjust our pH levels. Potassium ions. Creatine, which is a leftover product. It's a waste product from using our muscles. And certain types of drugs get secreted out this way. Okay, this is why urine samples can be used for drug testing. Okay, and this is going to be the last place where we can help regulate the pH of our blood, which is very important. So as we're coming out that distal convoluted tubule, this is going on. This sort of last minute adjustment. Wait, we meant to throw these guys away. Okay, let's make sure, is our pH all right? Okay, now we can say goodbye and excrete that urine. And then I included this. There's actually a great description and a pretty good picture in your book. There's a diagram in your diagram packet. I think it's diagram number 6 where we're going to look at this as well. But I just like that this was all in one visual for you, where it's showing you all the things that are forced out under that high pressure in step 1. And it's got a description up here. Then we move into the proximal convoluted tubule. And you can see water, glucose, amino acids, some things are getting reabsorbed, and it's got a description up here. And then after you go around the loop of Henle, you're in the distal convoluted tubule, and some things that didn't get secreted out before are going to get secreted out now. Some drugs, some ammonia, which is very similar to urea, some hydrogen ions to fix that pH. And then as it leaves through the collecting duct, any of that that's still in that urine is going to get excreted. All those things that got reabsorbed and the blood that's now freshly filtered is going to go back, leave through the renal vein to go back to our heart to re-enter circulation. Okay, one more time. This is going to correspond to page 521 through 524, and in class we'll be working on numbers 5 through 7 in our diagram packet, and I'll see you on Monday.